Alrighty. Hello, everyone, and welcome to Café Scientifique from all of us here at SFU's Faculty of Science. My name is Stevie Benish, and I'm the Manager of Outreach and Engagement here in the Faculty of Science. And just want to say welcome if you're one of our regulars. Great to have you back. And if you're new, thank you so much for joining us for the first time. This is actually our last Café Scientifique of our academic year, so we're so excited to see so many people have made it out tonight. For those who are new, Café Scientifique is a Faculty of Sciences public seminar series where our faculty presenters share their current research and its impact for our local communities and for larger society. We typically host about 6 to 8 of these a year. They're all virtual and they are all recorded. So if you miss one or want to share it with someone, or after you see tonight, you want to go back and check out any of our past talks, please do. We also love to hear from you and to keep in touch. So after the event, you'll receive an email with a link to join our mailing list, and so you can continue to receive updates. Just before we get started here, I would like to, start us off on the right foot by acknowledging that we are all privileged to gather on the unceded and ancestral lands of the Musqueam, Squamish, Tsleil-Waututh, and Coquitlam peoples. I'm coming to you from the SFU Burnaby campus. And I just want to say thank you for all of these people for having cared and being stewards for these lands and waters since before our times. We look forward to working with them in partnership as we continue to build and grow our communities together. So if you know whose territory you're currently on, I invite you to just take a moment to reflect on that. And if you don't know, this is a great time to learn. We have an incredible resource that we always like to recommend. It's called native-land.ca. And there you can familiarize yourself with all the Indigenous peoples whose lands we are so privileged to live and work on. So a couple little house rules, just very, very few rules here. Biggest thing is we hope that you ask lots of questions. We're going to have some Q&A time at the end. So you can ask questions using the Q&A box, and we will do our best to try and get to everyone's questions after the presentation. So anytime throughout the talk, please feel free to type your questions into that box, and we will get to them during the Q&A period. The chat will be turned off during the event, so just use that Q&A function. There is a live transcript, so if you need to access that, please do. And once again, as I mentioned, we do record all of these, so this will be recorded and shared on our website. So now, the reason we are all here, tonight's discussion. So I am so pleased to be able to introduce Dr. Dylan Cooke, who is an associate professor in biomedical physiology and kinesiology, as well as the director of the Sensorimotor Neuroplasticity Lab. I wrote this, I should be able to pronounce these things. So Dr. Cooke's innovative research program explores the link between the variation in brain organization and behavior, response to injury, and the brain's capacity to rewire itself. In laboratory experiments, variations between individuals has almost always been regarded as a complication to be minimized, and thus very little is known about these individual variations in brain organization. But Dr. Cooke aims to characterize and study the significance of this variation and determine how it affects skilled behavior, resilience in the face of brain injury, and natural adaptive changes in the brain. So I'm very excited for tonight's talk, where Dr. Cooke will be sharing with us how brain mapping gives scientists clues on how our brains work and how they've evolved. So over to you, Dylan.
Thank you, Stevie. I'm just getting my slides set up here. All right, welcome everyone. Before I start, I just want to encourage you to use the Q&A box if you have questions at the end, but also if you have a question about something I said that doesn't make sense or something on my slides that doesn't make sense, I encourage you to also post something in the Q&A box that's brief so that I can try to keep up with it, and just write clarification if you have a question about something that I said or something on my slides, and I'll try to answer it as we go so that I don't lose anyone along the way. Also, feel free to put longer questions or sort of bigger picture questions in there, and we'll save those for the end. And I really encourage you to think about questions. I hope this is a thought-provoking talk. I'm also going to make a brief land acknowledgement here. I'd like to gratefully acknowledge the unceded territories of the Tsleil-Waututh, Coquitlam, Squamish, and Musqueam nations, where our campus is located. This land means a lot to me. I work here, I live here. I raise my children here. I commute through the woods here. I grow food here. And I give thanks to the people who have cared for this land, and I will try to do my best to help care for it too. So generally today, I'm going to be talking about brain mapping, and I'll explain what that means. Um, we're going to start off thinking kind of bigger picture about what brains are for, and even where the brain is in the body, how we know that. And then we'll sort of zoom in and talk about mapping functions to brain areas, mapping within brain areas, mapping single neurons, which is single brain cells, and it's mapping their function. And then we'll get to the work that I do here, which is motor mapping, and talk about individual differences in brains and muscle maps. I'll explain what that means. If there's time, I'll talk about my work on echolocation control in bats. And then I'll finish briefly with the sort of future of brain mapping. So before we tackle brain mapping, let's think about what brains are for and what they are. So one thing that brains are not, they're not necessary for life. This seems sort of counterintuitive, but most organisms on Earth today, and most organisms that have ever lived on Earth, do not have brains. This is the tree of life, showing species, showing species branching out from the last universal common ancestor, or LUCA, that lived about 4 billion years ago. And as you move forward in time, you move outward from LUCA out toward the branches of this tree. And these are all of the groups of living things today. Most of them are microorganisms or plants. And a very small number of us are animals. So all of these ones with the red bracket do not have brains, and they've been doing fine for 4 billion years. Thank you very much. In fact, there were no brains on Earth until about 500 million years ago. So there's 3 billion years at least of life on Earth without brains, and it did very well. Brains are very specialized for a particular kind of living thing, and that is most animals. So let's zoom in on animals, which are the only living things that have brains. So here's our tree of life again, and we're zooming in on animals, and we're exploding this out here. And again, this is a sort of extended tree of life. And we are here. Humans are mammals, and mammals are vertebrates. And so we are in this group right here. We have brains. And insects and shellfish and worms have brains. Some animals, like starfish and jellyfish, have nervous systems, but they don't have a brain. And there are some animals, like sponges, that don't have a nervous system at all. So all of the animals that are inside this green box have a brain. And brains first appeared again around the time of the Cambrian explosion, about 500 million years ago. And my own work is in mammals, which again is in this group right here. So what are brains for? Now that we've done some comparative neurobiology, looking at which living things have brains and which don't, let's think about what brains are for. The animals that have brains, most of us animals that have brains, or nervous systems, we move relatively quickly compared to things like plants and in response to things in the environment. So brains make this happen by gathering sensory information, taking in sensory information like what we see and feel and hear, And then we process and compare this information. We combine that information. And then relatively quickly, we make relatively fast, coordinated responses. So let's take an example. Imagine you see an apple hanging from a tree and you process this information. You see that the apple is red and you process that to know that it's ripe. And you also process where relative to your body that that apple is. And you use that information to coordinate the different parts of your body together, again, relatively quickly compared to plant. You move your feet to walk over. You reach your arm out. You grasp with your hands, and you bring the apple to your mouth and you bite. So that's muscles from all over your body that are being coordinated within a few seconds to bring some food to your mouth, and you can take that food in. Uh, you compare that to a brainless organism like most plants. Most plants can move relative to the sun, but it's for most of them, it's a lot slower. And most of the animals in, or most of the living things in the world are microorganisms. And they're only one cell or a few cells. And whereas when we walk across the ground to pick an apple from a tree, we're coordinating billions or trillions of cells together to make this movement. So that's what brains are for. It's for making relatively fast, coordinated movements, and it's something that animals specialize in. So the first stage of brain mapping is to map where is the brain in the body. And by brain, I'm talking about this brain function that I just explained, the idea of there's something in our body that is integrating information from the outside and processing it and allowing us to coordinate across our body a response to that information. Now, consider that it's really not obvious where in the body this function is. People have always understood, any person that butchered an animal, for example, would understand that there's something inside the animal's head. And they might even, encounter a dead person and they might dissect it and find out that there's something inside their head, this soft and squishy thing. But they wouldn't know, and there wouldn't be any clues from that really about what the brain function is for. In fact, um, even a couple of hundred years ago, there were theories like, oh, the brain is, and this is in Western medicine, the brain is an organ for cooling the blood because the bumps on the brain looked like the bumps on a radiator. Um, and traditionally, across many cultures all over the world, there's been an assumption that brain function was not in the head. Um, so, for most cultures, it was thought to be in the heart. And one example of this is in ancient Egypt. In ancient Egypt, we have a lot of evidence about how the body was viewed through mummification. And the first step of mummification was to put an iron bar up through the nose and scoop out the brain of the dead person and remove the brain and throw it away. In contrast to that, the organs in the chest and especially the heart were placed in special jars and saved or were placed inside the body in these jars. And this and other writing it confirms that the ancient Egyptians and many other cultures didn't think the thing that's in our head was very important to us. It wasn't the seat of the intellect, and it wasn't the thing that controlled all of our movements. That they ascribed to the heart. Now, despite this misunderstanding that many cultures have, there was evidence, even a long time ago, that brain function is in the head. In fact, some of it comes from ancient Egypt. So, for example, head injuries, evidence of head injuries in the form of this papyrus, which is a field guide for battlefield surgery, written about 5,000 years ago. This papyrus talks about the fact that a soldier would have an injury to a particular part of the head, and then as a result of that, they would walk with a limp. And they're making a connection there between the control of the control of movement of the body with something that's happening in the head. And in fact, this papyrus has the first written instance of the word brain in any language. It's used multiple times in this battlefield surgery guide. Now, there's another early piece of information, early clues about what people thought about brain function and where it was. And that is the practice of trepanation. So trepanation is skull surgery in which a stone or metal tool was used to make a hole in the skull. This is the first neurosurgery, and we have lots of evidence of this all over the world, in cultures all over the world, dating back as early as 10,000 years ago. Amazingly, we can tell from the scar tissue that formed around these holes that up to 90% of the people that received it, sometimes it was less, sometimes 50%, but up to 90% in some cultures survived. If they died from this, there wouldn't be time for the scar tissue to form around this hole in the skull. And so we know that many of the people survived this, which is sort of amazing before the current understanding of how to do surgery in a hygienic way. Now, for most of these skulls that we have that show trepanation, we don't know why the operation was done. We do know in a few cases, there are instances as early as the year 150 of the Common Era, so that's more than 1,800 years ago. There are instances where we know that people were doing it to treat epilepsy. And if any of these earlier cases, again, that go back as early as 10,000 years ago, were also to treat epilepsy or mental illness, is another one that we know that happened later, either of those things would be evidence that the people doing them understood that these functions took place in the head. Epilepsy might be evidence that they understood that the brain controlled the movements of the body. And if it was to treat mental illness, then it would be to understand that our thoughts and emotions were processed in the head as opposed to in the heart. So again, it's interesting. We don't know for sure, but it is possible that there's evidence as early as 10,000 years ago that there was an understanding of where the brain is in the body. Now we're going to fast forward a bit, heading toward the present, but still a ways back in time. These are examples of mapping functions to brain areas. Some of these you may have heard of before. I'm going to go through these relatively quickly. One was the case of a man named Phineas Gage, who was injured in a railroad accident when an iron pole that he was using to tamp down dynamite set off the dynamite and exploded through his head and left a hole through his face and through his brain, damaging part of his frontal lobe. And afterward, he was described as having changes to his personality. And so there was a link between the personality and mental abilities like planning that were damaged in his case because his personality did change when his frontal lobe was injured. Another famous example is the case of a man in France in the mid-1800s who had a brain injury and lost the ability to produce speech. He could still understand speech, but he couldn't speak himself. And this was understood by a scientist named Broca. He interpreted this in the case of another patient, that this place in the brain was related to the production of speech, the function of speech. And then the final example here would be the case of a patient who was known by the initials HM, who had epilepsy and had a surgery to remove part of his brain called the hippocampus. And he was one of the first cases in which this surgery was done. And he suddenly lost the ability to form new memories. And this led to a large number of studies, many of them done by a scientist named Brenda Milner, who is still alive at the age of 106, who did her work at McGill University. and taught us a huge amount about how memories work in the brain and where they are, where that function is in the brain. Again, all of these are examples of how brain damage led to better understanding of the particular damaged areas and what their functions were in the brain. One of the earliest cases of mapping within a brain area came again from brain injuries. And I'm going to start with the story of Tatsuji Inoue and or Inoue. And he was an army physician in Japan at the time of the Russo-Japanese War. And there was a switch at this time that happened where armies started to use different kinds of rifles that fired much smaller, much faster bullets. And these bullets, maybe half the diameter of the bullets that were used, for example, in um, the Franco-Prussian Wars or the American Civil War, um, were again about half the diameter and traveled almost twice as fast. And the result was that the bullets would go into the brain and travel straight through the brain and exit the brain without leaving giant holes in the skull. And people were able to survive these better, especially with improvements in battlefield medicine. Um, and so Tatsuji Inoue's job as this army physician was to measure when bullets went through the part of the brain that controlled vision, how much were the soldiers blinded? How much of their whole visual field were they blind in? Because that determined how large their pension would be. The more that they were blind, the larger the pension. Um, he was extremely observant and careful, though, in addition to measuring essentially the blind spots in their field of view, he also measured where the bullets entered and exited the skull. And from that, he was able to figure out exactly which part of the brain had been damaged, assuming that the bullet traveled in a straight line, which it usually did. And from this, he was able to determine that um, when a when a bullet would uh, when a bullet would damage a particular part of the back of the brain, which we now know as the primary visual cortex, that the blind spot would always be in a corresponding spot in the visual field. So this is a map of the visual field. This is the upper half, and this would be the right visual field. And this is in close to the center of vision, and this is further out away to the edge of vision. And I've colored these lines to correspond to um, where in the brain the same region of visual space is recorded. So your center of vision, which is right here on this diagram, was represented or processed at the back of the brain, on the inside, right near the very back pole of the brain. And when in cases for soldiers that had blindness in their periphery, so out on their peripheral version out here, that would be further forward in the brain. And so it's almost like a latitude and longitude. You could predict based on where the bullet went into the brain, where the blindness would be, or vice versa. And he mapped this in many cases, and realized that there was what's now called a retinotopic map of the brain. It's mapping the retina, which is the part of the eye that captures light, precisely onto the surface of the brain, in the sort of back middle of the head. And the same thing was done a few years later by a neurologist, Irish neurologist who worked at a British field hospital in World War I named Gordon Holmes. He was faced with the same thing, again, small, high velocity bullets. Many of the people survived. Many of them were blind in very specific places. And they had very specific injuries to a specific part of their visual cortex. And so this being one of the first times that the organization within a brain area was mapped led to the understanding that information that is processed uh, information is often processed near similar information, so that we understand now that our brain processing one part of space often wants to compare that part of space to the adjacent part of space so that you can use that to recognize a shape that might be in both of those parts of space. And the best way to do that is to have an exact map of the visual field in the brain where adjacent places in your visual field are in adjacent places in the brain. And so you end up with these beautiful maps of exterior sensory space. And this is true for vision, and it's true also for touch on the surface of the body. There are maps of the body inside the brain. And this also makes it extremely convenient for scientists because it gives us a way to study other phenomena in the brain. So this was one of the early cases of mapping within a brain area. Okay, and I don't see any questions in the Q&A so far. But again, if there's anything that doesn't make sense, please put it in there. So far we've talked about what brains are for and where the brain is in the body, and then mapping functions to brain areas and mapping within brain areas. And next I'm going to talk about mapping single neuron function. So we're zooming in more in space. And this is how I got into neuroscience originally. So my life changed in a small but important way when around the time that I was starting university, I learned that the brain constructs perception. Now, what does this mean? So I realized that my whole experience of the world, including vision, was not real, but just a very good approximation of it in my head. And some people, this doesn't matter to, but for me, this really was a very deep thing to absorb. And one example of this is that optical illusions took on a deeper meaning for me because optical illusions like this one here, this is the famous Kinesiology triangle, which is three sort of Pac-Man shapes and three little angles here arranged in a way that our brain interprets as there being a white triangle laid on top of and blocking some other objects, three circles and an open triangle here. Um, this perception gives the impression that there is a white line, a white edge right here that is not there. And so optical illusions are great at revealing these cases where the brain is constructing a reality that doesn't exactly match the reality in the world. And optical illusions work because they toy with these systems a little bit. So I became very curious about how the brain turns light, for example, entering the eyes into perception, how it builds perception from light entering the eyes. So in my second year of university, I took a course on visual neuroscience, which gets at exactly this question. And so I learned about a few of the experiments I'm going to tell you about right now, in which scientists studied the behavior of single brain cells in the primary visual area in a cat brain, which is similar to the human visual area in important ways. And by moving a spot of light on a screen in front of this cat, they located the place in the cat's visual field that was monitored by a single neuron. So what does this mean? Let me try to demonstrate what this means. So they're shining a light in different places on this screen here. Oops, my mouse is a little bit jumpy, so I'll move slowly here. They're shining a light back and forth here, and they are recording the activity, or they're amplifying the activity of a single neuron in the cat's brain. And whenever the spot of light moves into this area that is shown in green but is not actually there, then that cell's firing rate would increase. So you'd hear it's played on a loudspeaker, and the scientists could actually hear a sort of pop, pop, pop of this cell firing. And so they're able to mark on the screen where this was. And I'll show you an example of that. So here's light, a circle of light on the screen, and they move that light across the screen, and they record a few pops of this neuron, a few action potentials of this neuron. They didn't get great responses from this neuron, but by accident one day, they discovered that if they used a different shape of light, if they used a bar of light instead of a circle of light, and they did the same thing, they got a much larger response. All along, they'd been using circles of light because that's what everyone had used before, and they used this bar and they got a much better response. And then they discovered that the angle at which this bar was presented on the screen strongly affected the response of the neurons. And so the best response for this particular neuron was a bar of light at about 10 o'clock, aiming at about 10 o'clock. And then as they moved away from that preferred orientation, the cell didn't fire as much. So then they did additional experiments, and they found a pattern in the map of which angles neurons preferred. So here's an example. Here's a piece of brain right here. And again, sorry, my mouse is very jumpy right now, so I'm going to try to move slowly. And they moved an electrode over about 9 hours. They moved it slowly, diagonally across the brain, because this is straight up and down in this piece of brain right here, this cortex. So they're moving diagonally across the brain, and they tested 53 neurons over about 9 hours. And these are the preferred angles of these 53 neurons. You can see that the preferred angles change very gradually as they move along this trajectory across the brain. And when they plotted it here across the distance they traveled, the preferred angle decreased and then it increased, but it moved very smoothly. And so what they came to realize after a while was that here's this map of visual space in the brain that embedded within that or coexisting within that map of space, visual space in the brain, is also a map of orientation in the brain. Then many experiments later, they and others figured out that in addition to space, visual space, around just the space that you see, that's what Inouye and Holmes figured out from the bullets in World War I, the Russo-Japanese War, and orientation, which was these bars of light at different angles. There are additional maps that I'm not going to talk about today for movement, for depth, and for color that are all embedded within this same map of visual cortex. So you can see here, orientation is varies across here. And all of these other things vary within this sort of cube of cortex. And this is like 1 pixel in your field of vision. And all of these things are very systematically monitored by your brain. Now, the actual shape are not exactly cubes. Further advances in the way these experiments were done showed that they're actually sort of pinwheel shaped. And so this is, so instead of just a cube, you can see that the orientation preference of neurons makes a sort of swirling pattern across the brain. Now, what is the point of all of this research? I want to note here that most of the experiments that I'm going to talk about today, all the ones I've talked about so far on single neurons, and a lot of the ones coming after this, are so-called fundamental research or basic research, which means that they are not designed to cure a specific disease, but instead are curiosity-driven And they aim to lay down a foundation of knowledge that others can build on. And it's too long a story for this talk, but this work did lead directly to discoveries that quickly changed the way that eye surgeries and other interventions were done in babies with visual problems. And this saved many of them from losing their vision. So basic research is an essential part of science, and it often leads to unexpected and useful discoveries. So all of the single neuron visual mapping that I was just talking about, and the studies with the bullets in World War One, all of that was looking at the primary visual area in the brain, also called V1, and I've highlighted that here in this monkey brain. And this is one of dozens of other connected visual areas, some of which are shown here. There's many more than this in the monkey brain and in the human brain. So I was learning about all of this in this second year neuroscience course at university from this professor, Charlie Gross. This was my professor for this class. Now, his work was on the other end of the visual system. So I was showing you work in V1 cortex. He was working in a brain area that's called IT cortex. And I'm going to tell you a bit about his work. This is also something I learned in his class. So this is a part of the brain that processes complex stimuli. Instead of just dots and lines, he showed that single neurons in this brain area would respond to a specific monkey face, or often they respond generally to all monkey faces. In this particular example, also responds to pictures of human faces. It even responds to a cartoon face. It doesn't respond to a hand. It doesn't respond to a cut up picture of a monkey. It responds sort of weakly to a monkey or a human with the eyes removed. So they did lots and lots of comparison experiments, and it seemed like the most likely thing that was going on is that these single neurons are specialized for the processing of faces, which is an incredibly important thing for humans and monkeys because we're such social animals. So I found this just amazing. This was for me, this was really kind of the answer to this riddle that I had been curious about how does the brain turn light into and then build up forms and shape into complex shape like faces that have meaning, emotional meaning to us. And by looking at a single neuron at a time, if you string all of these experiments together, you begin to understand how the information coming to the brain is turned into a meaningful image like a face. We don't have all the pieces even yet today, but this sort of gives us a pathway to understand how it is that the brain does this amazing thing. So I really wanted to do this. Was soon volunteering in Charlie Gross's lab. This is Charlie Gross. I was volunteering in his lab, and I got involved in experiments in the motor system, which controls muscles, and I'll talk about that soon. Now, about 2 months after I joined his lab, Charlie invited me and all of his students up to his summer house, which was about a 3-hour drive north, driving past New York City. Now, none of us had a car. So Charlie, who was going up a few days before, drove up, but he gave us the keys to his other car so that we could drive up together and join him. So the other students and postdocs in this car either didn't have a license or they didn't want to drive in New York City. I didn't either, but I ended up behind the wheel. And this is before GPS, and so we took a wrong turn at some point, and then we're on the Cross Bronx Expressway heading completely the wrong way. And I come around a corner and traffic has stopped and I don't stop in time, and I rear end the car in front of me. Now, no one was hurt, but I was mortified. I just crashed my advisor's car. And I didn't know what I was going to do. I idolized this guy. I wanted to work in his lab, and I'd crashed his car. How could I be in his lab if I'd crashed his car? I felt terrible. So Hours later, we arrived. We pulled up at his summer house and explained what had happened. The license plate was wired onto the front bumper. Um, and to my immense relief, Charlie was really gracious about it. And in fact, I continued in his lab um, for 2 more years as an undergraduate, and then I did my PhD in his lab as well. And in fact, this is a few years later. This is me as a grad student in his lab. I'm trying to catch up to his hairdo. I'm not quite succeeding. Um, so uh, uh, this the work that Charlie taught me about and the work that he did were really important in getting me into the career of science. So here's a bunch of us on a hike near Charlie's house. Again, this is a few years after the car crash. Um, Several of us here, the ones I've named here, were involved in this motor mapping experiment. I'm going to tell you about that now. So in this case, um, we're mapping the brain not by listening in on the activity of neurons. We're actually injecting a small amount of electricity through an electrode into the brain. And this electricity is traveling through existing circuitry in the brain. It eventually reaches the muscles, and the muscles contract and they create movements. And so this is a way of sampling different places in the brain with an electrode. At each place, you zap the brain, and then you see if there's a movement of the body and what that movement is based on where the electrode is in the brain. So one of the things that we found, I'm going to go through this pretty quickly. One of the things that I found, that our group found, is that complex movements, like whole movements that looked like a behavior, were coded at single locations in the brain. By coded, I just mean that one spot in the brain was associated with a particular movement. So, for example, we'd put an electrode in at one of these red locations, which is about here on the monkey brain. And we'd stimulate at this location, and the monkey would make a flinching movement. And at other locations, we would stimulate, and the monkey would reach out and grasp something that wasn't there. Or at these pink locations, the monkey would make these multi-limb climbing and reaching movements, or they'd make movements like they were trying to manipulate something with their hands, or we'd stimulate and the hand would form into a grip like it was holding food, and it would move to the mouth and the mouth would open like this. All of these were in different locations. And so it seemed like we'd found a map of sort of behavioral space in the brain. And I'm not going to talk in detail about this today, just to say that this led to the experiments that I'm doing now. I'm going to take just a quick aside here to say that there's been so much mapping in brains of different experimental animals and in people that we now have what are called brain atlases. These show the exact X, Y, and Z coordinates of each brain area. And this is one page from a mouse brain atlas. Um, and This is a coronal section, which is a cut sort of like this through the brain. And maybe there'd be something like 100 of these pages, each one at a different place, front and back in the brain. And then there's a whole other chapter that's all on sections that run this way across the brain. And then a whole other chapter that's sections that run this way across the brain. These are incredibly useful. Even tiny little brain areas in the middle of the brain are located on here. These are a great tool. There's a problem, though, and that is that this is a sort of average mouse. Um, and Not all mice are the same size or have the same size brain. And in fact, even if you take two mice that have the same size brain, these brain areas are not exactly all the same size and shape within the brain. They're roughly in the same position, but not exactly. And so there's this problem of individual variation that that has sort of become a theme of my research. It's something that I saw in my own experiments. This is a different experiment. This is an early motor map in rats, and it's just zoomed in on the part of the motor map that controls the rat's paw. And um, the different color, don't worry about the details, but the different colors are different movements in different directions of different parts of the paw. And what you'll notice is that across these three rats, the coloration shows you that these maps are radically different from each other. These are three rats, same species that are brought up in the same cages. And these maps are really, really different. In the original paper, these were in black and white, and they were all in different pages in a 20-page long paper. And the authors of this paper on page 18 mentioned in one sentence that, oh, these maps were kind of different from each other. But generally, no one doing these experiments wants to highlight how variable their data is because it sort of makes you look sloppy. I mean, it's not really. I mean, this is the reality of brains, is that they vary a lot. But generally, people don't talk about this. Um, and I actually don't see this as a problem. I see it as an opportunity, and I'll explain why a little bit later in the talk. Um, but when I realized and I was setting my own lab here at SFU, and I realized that I wanted to understand individual variation. I study motor maps, which are especially variable, and I wanted to really zoom in as much as possible on these motor maps to try to figure out um, exactly which things vary between individuals, and then I'll figure out later what does that variability mean. Oh, and I see here a few questions here. Have you used the virtual brain in your work? I haven't. I know about it. Some of my colleagues were um, helped create that. And it's possible that some of our data will eventually go into virtual brain. This is a sort of a way to model brain data. Um, I'm just looking at these other questions here. Uh, there's some good questions that I'm going to get to at the end here. Yeah, great questions for the end here. And one question that I will answer now, does the brain have similar components to a circuit? Sort of. Um, we often talk about brain circuits. Brain circuits are made out of different parts than a computer circuit. There are some similarities, but there's some really important differences, and that's a sort of a big discussion. But so far, I'm going to come back to these other questions later, if that's okay. I'm going to move on. So there's time to get through a little bit more. Okay, so my lab here at SFU uh, studies the brain organization of rat motor cortex. This is the part of the brain that controls and coordinates movements, which means it's controlling muscles. So to do this, we start with anesthetized rats, and we record electrical activity from muscles. And this is electrical activity from muscles is called EMG, and you'll see that on some of these figures. And we electrically stimulate different locations in the brain. You'll see ICMS a bunch of places in these figures. This stands for intracortical microstimulation. This is just a fancy name for brain stimulation. So brain stimulation mimics natural electrical signals that are in the brain, and it activates circuits. It causes EMG in muscles, and the EMG in muscles causes movements of the body. So we do these experiments and we move the electrode around to different places to make our map. So the first thing we do is in different places, we observe different movements of different body parts, and we analyze which body parts move, and then we make a map of the brain. We use this color code here. So we color different parts of the body or different joints of the body, different colors. And then this here is a side view of a rat brain. This is the rat cortex on one side, and this is the part that we are studying right here. And I've zoomed in on this part of the rat cortex. And we're looking at motor cortex and also some adjacent parts of the parietal lobe here. And this has been color coded again like this. And so blue and purple areas are movements at the face. Green and yellow are movements of the forepaw, like what we'd call the hands and arms. And red is movements of the hind paw, the hind limb, what we would call the leg in a human. Now, you'll see that a lot of the places in the brain where we've stimulated the brain, we are creating movements of multiple body parts. That's why we have these stripes here. So red and green means that we have movement of the leg and also, in this case, the elbow. It's an elbow movement and a leg movement together. And sometimes it's even more than that. If we just zoom in on one of these sites, now remember, we're recording from muscles, recording activity of muscles. And so at this one site where we get movement of the leg and the arm of the rat, so to speak, we had activity of a dozen different muscles. These are all different muscles, and this is the activity of the muscle, the EMG activity during brain stimulation. And then if we zoom in on a few of these, this is the period of brain stimulation, this gray bar right here. And you can see that different muscles turn on and off at different times. And so we're not just seeing all the muscles contract together and then relax when the stimulation is done. Instead, we're seeing a sequence of muscle activity that mimics natural movements where different muscles turn on at different times. If you need to reach out, your arm needs to be accelerated. And then when it's about to grasp something, it breaks. The body actually slows down the arm, and then your hand might open to grasp as you approach the thing you're going to grasp. And then when you land on it, then different muscles are going to cause you to grasp onto the object. So we're seeing patterns that look sort of like natural behaviors. It's not that surprising given the other work that we've done, but now we're seeing it down to the level of individual muscles. So we can make maps. This is a map that I showed you before of movements in the brain. We can also make maps for these individual muscles that we're studying. So for example, this shape right here in the brain, if we replace the movement data with the data just from the biceps muscle of the rat, then we find the biceps is controlled in this part of the motor map. And so on. These are the maps of other muscles. I'm going to kind of zoom out here and show you a huge amount of data at once. Don't worry about the details. I just want to show you the kinds of analyses that we're doing with this. So this is the muscle map for one muscle, the lats, and this is a muscle map for a different muscle, the trapezius. And this is all of the parts in the brain where when we stimulated, we activated both of these muscles together. So it's what we call a coactivation map, or you could think of it as a coordination map. These are the parts of the brain that coordinate these two muscles. Now, this is all of the different pairs of muscles that we've studied. So in black here, these are the individual muscle maps. And then you can take any two of these, like this one and this one, you can find where they intersect. And that's where in the brain is the coordination of those two pairs of muscles. The details here aren't important, except to say that almost all of the pairs of muscles going from the face to the hind limb, there's somewhere in the brain that's coordinating that pair of muscles. So almost all of these little brain outlines here show some activity somewhere. This makes sense because there are times when we need to coordinate very disparate parts of the body. And if you think about it, when you move, you're almost never moving one muscle at a time. Just to reach your arm out to grasp something, you're going to use dozens of muscles. And so almost all natural movement is a coordination of muscles. And we're starting to map coordination of muscles in the brain. That's the goal here. Now, these are the movement maps, like the first ones I showed you, just showing which joints of the body are moving. And these are movement maps for 6 different rats. And what you'll see is that there are some patterns that are similar across these 6 rats, but there also are some huge differences between these individuals. We're still at the part of our research where we are figuring out which things vary and how much they vary. We haven't yet figured out what this variation means, but I want to tell you, looking down the road, what we're hoping to do with this information. So we're interested in figuring out if um, if there's a particular pattern in the rat brain that is found, if we find that pattern in a rat, does that mean that that rat is going to be better or worse at learning a particular skill, like grasping a food pellet is a classic one that rats are trained to do. Um, and maybe that rat will be better at this task, and a different rat that has a different pattern will be worse at that task, but better at a different task. Um, if this is the case, then in the future, we could potentially do brain scans in people and use the information about their own personal brain organization to tailor the way they're going to be trained to learn a sport or a skill, whether it's tennis or surgery or playing the piano. It may be that there are organizations in the brain that mean that you learn in a different way. It could also mean that we could tailor rehabilitation. If there's some sort of injury, it might mean that you should be rehabbed in a particular way and not in another way. We're way before even figuring that out, but that's the sort of eventual goal. Another potential goal we have is to see if there are particular brain maps in rats that when there is brain damage to that rat, there is more or less effect on the way they move. So if you simulate a stroke in a rat where part of their brain is disrupted, um, then do they have more trouble reaching for a food pellet? And do they have less trouble reaching for that food pellet if they have a different pattern in their brain? If that was the case, what we hope is that we could then figure out how to uh, give people who are at risk for strokes particular coordination exercises to rewire their brain to make their brains more robust in case they do have a brain injury. So if they're going to have a brain surgery or if they have a family history of stroke, then maybe they should do these coordination exercises so that their brain is rewired to a particular pattern that we know that in rats protects against stroke, and that could potentially protect people against stroke. Again, we're far away from doing that, but that's the sort of long-term goal of the direction that this research is headed. And there's a lot of other interesting questions that we can ask about individual differences in brains and the way the brain works that we're hoping to focus on in the future. Okay, I do not have time to talk about bats, so I'm going to fast forward through that um, and finish with this. So, looking beyond my own research, there is a very different way of thinking about brain mapping that has opened up in recent years. And this could be not the future of brain mapping, but a new future of brain mapping. And um, what it is creating a connectome. Connectome is analogous to like a genome. A genome is mapping all of the base pairs in our DNA and then figuring out exactly what versions of genes we have. And it's been hugely important for genetics and medicine and drug discovery and lots of other things. Connectome, instead of the pattern of base pairs in our DNA, a connectome is all of the connections in a part of the brain or the whole brain. And in this study that came out about 6 years ago, um, this group did an impressive connectome of a teeny tiny part of a rat brain. This is like you can imagine the size of a grain of salt. 1/1000th of that. That's the size of this connectome. But in this connectome, there were something like 90 neurons and meters of axons. And they mapped every single one of these, and it took a team of 100 people 2 years to do this. A huge, huge amount of work. Um, And just this month, about 3 weeks ago, a different group came out with something similar, but much, much bigger. Um, this is a consortium across universities, and they mapped uh, a volume of the mouse brain that was about 1,800 times larger. And they've mapped every single one of the 80,000 neurons that is in this chunk of brain, about 4 kilometers of axons, and about 500 million synapses. And the idea is that eventually they want to do this for the entire mouse brain and eventually the entire human brain. And once you know every neuron and every connection in the brain, you could learn a tremendous amount. Now, I'm not worried this is going to put me out of business because, first of all, at the current rate that they can do this in mice, it's going to take them several hundred years to do an entire mouse brain. I think it will be faster than that, but with their current technologies, it would take hundreds of years to do an entire mouse brain. Even once they do an entire mouse brain, there's still going to be a tremendous amount of work to understand what this huge amount of data represents. So I think this is going to be a tool in the way that the Human Genome Project is a tool. It doesn't end genetics. It's just a tool that geneticists can use. And this will be a tool that neuroscientists can use to understand the brain. And it's at a part of its history where it's expanding rapidly. And I think this is going to get really big in the next, say, 10 years. So watch out for connectomics as sort of the future of brain mapping. Okay, I need to finish up here and start taking questions, but I just want to say thank you to the people that did this work. These are my collaborators that worked on the bat work that I didn't get a chance to talk about. These are students in my own lab, my PhD student Milad, and James and Saba are some of the many undergraduates that have worked in my lab. These are some of the other ones that have done great work in my lab over the last few years doing that rat work. So thank you for your attention, and I look forward to your questions.
Wonderful. Thank you so much. That was fascinating. Yeah, and I've already taken a look at some of these questions, and they are equally as interesting. So let's get into them. So the first question here is, you emphasize the function of a brain when it comes to interaction with the outer world. But I think one of the biggest functions of the brain is abstract thinking, which doesn't seemingly have anything to do with the environment. So yeah, do you wanna?
Yes. So abstract thinking is really important, and it's a lot harder to study. Some people do study it, but it's a lot harder to study in animals because you can't ask animals what they're thinking. And so often when you're doing work like mine that's invasive, that involves surgery on animals, you're going to choose a part of the brain to study that's easier than abstract thought. Now there are some people that do very clever work where they are train animals to count things, and so there are people studying things like numerosity, the understanding of numbers in animals, and then they do experiments in their brains to understand where this is coded. So it's not that it's impossible; it's just that it's more difficult, but it's super interesting. So, um, but yeah, I guess my main point here is that if you want to understand abstract thought, then you either work with humans or you train an animal in a very clever task. If you want to understand a sort of general principle like individual variation, then you choose something easier, like the control of movement, because you don't have to train an animal. You can just look at the movement and then you sort of have your output.
Great, thank you. Alrighty, our next one here is, if perception is directly related to consciousness, would the origins of perception give hints to the construction of consciousness?
Consciousness is actually one of the things that first got me interested in learning about neuroscience, and it was this idea for me about this sort of when you accept that uh, the reality around you is constructed in your brain, and that consciousness again is not your sort of awareness of your surroundings, which includes vision, is not uh, it's not outside of you. It's something that again comes from your brain. Then you have to figure out if you're a scientist, you want to figure out, well, where in the brain is consciousness? And there are a lot of people working on this problem. It's difficult to study. Um, and let me give you an analogy about this. Um, the if you're studying vision, um, and uh, you have the retinas in your eyes, and then there are nerves that goes from your eye to the middle of your brain, and from the middle of your brain to the back of your brain, where primary visual cortex is. You can cut those nerves, and then you will become blind. If you cut the nerves for one eye, you'll become blind in that eye. Does that mean that your vision is processed at the place that you damaged in the brain? No, it just means that the place that you damaged is part of a system. It's a necessary part of a system for seeing. But the one place that you cut that nerve is not the only place that does vision. And you have a similar problem for consciousness. You can have someone with a brain injury, like maybe earlier on it would have been a bullet, a bullet to the head during the Russo-Japanese War, and they might be unconscious, but otherwise their brain function might be in good shape if you could somehow test it. Um, but that doesn't mean that just that one spot in your brain is the place where consciousness takes place. Most likely, consciousness, like the visual system, is a very diffuse network that has many different parts. Um, and so, if you lose one of those parts, you might lose a part of consciousness. And there's some really interesting studies where people lose their ability to be conscious of a particular part of space around their body. It's really weird and fascinating. You can read, for example, books by Oliver Sacks to learn about this. He's a great author, um, Sacks, I think. Um, uh, and so there's lots of interesting case studies in this, and a lot of them have to do with perception. Um, but there's this frustrating problem that we don't know. We don't know a spot in the brain that is where consciousness is coded, and the consensus seems to be that it's probably a whole network.
Great. Thank you. Already a little bit less heady, hopefully, but maybe a little bit more straightforward question now. Can Alzheimer's and other brain-related diseases be mapped similarly to what you're doing?
So yes and no. So Alzheimer's is not my field, and you can't map it in the same way that I've described mapping. But because I mean, what I've been talking about is mapping responses to visual input or mapping motor outputs. So instead, when people talk about mapping something like Alzheimer's, you might figure out, here is a particular chemical or protein in the brain that is correlated with Alzheimer's, and then you can figure out if that chemical can be replicated in an animal. And then you can do a study where you try to stain the animal's brain um, exactly where every place where that protein or chemical shows up. And then you can create a map of where is that protein, or if it has to do with the expression of a gene, there are maps of gene expression in the brain. And that can be a way of mapping where a particular disease is in the brain. So absolutely, yes, um, that can happen. And by the way, there are some really cool techniques now where you can do that. And the traditional way of doing that is that you stain for a particular protein. So all of the proteins associated with a particular disease might be stained a sort of glowing green. And then you would slice the brain up into 100 sections, and then you'd look at each section, and you try to reconstruct where in all of these little thin sections of the brain this glowing green chemicals are. There is a relatively new technique in which case you leave the whole brain together and you soak it in chemicals that make it clear. And then you can look at the brain from different angles and photograph it with a microscope from different angles without having to cut it up in the first place, which is much more it's a lot better, it's a lot easier in some ways.
And so this is another way that you can map something like Alzheimer's or disease. This is called clarity. It's a way of visualizing a brain without cutting it up.
That's really cool. Um, Alrighty, so our next question here, I think going back to a little bit earlier in your presentation, is about how do they differentiate between the different faces?
Um, so I'm assuming they're asking how does the brain differentiate between different faces? And we don't know the answer to this, but there's been a huge amount of study and thought about this. And before my advisor, Charlie Gross, found these face cells, there were people talking about this problem. And the problem is called the grandmother cell hypothesis or the grandmother cell problem. And the idea is if we know that the brain can discriminate faces, you can identify your grandmother, you can identify your friend, you can tell the difference between them just by looking at their face from any number of different angles. So we know the brain can do this. How does it do it? Well, one way that it could do it would be that it would have at least one neuron for each person. Like one of the cells in your brain might be your grandmother's cell. And then another cell might be your uncle's cell. Another cell might be your brother's cell. Um, and this was sort of a thought experiment, but it led to the realization that there was a problem with this, and that like, well, what if that one cell died? Would you forget your grandmother? And also, the number of people, that you can identify from lots of different angles, and the number of objects you can identify is enormous. Do you have enough space in your brain to have one neuron for each of these people or objects? And so the more likely thing is that uh, you have um, multiple neurons, a sort of combination of neurons, right, might represent your grandmother and different aspects about her. And that some of those same neurons might also help you recognize someone else that has similar eyes but a different mouth. Um, there have been cases where people have found there was a study that was about the Jennifer Aniston cell, where they were showing pictures of celebrities, including Jennifer Aniston, and finding single neurons that seem to code a particular person. But the problem is that there are, billions of people on Earth, and you can't show all of those pictures while someone is in an experiment. So it's a very difficult experiment to do. And you don't know if those cells might also they might also code a monkey face or a dog's face or something like that. So um, they're difficult experiments to do because there are just so many different objects that your brain can recognize and discriminate. And there are people working on this right now, and we don't know the answer yet exactly how it is that we can discriminate faces. We just know that the brain can do it.
And then we have a follow-up question, um, and that is that then related to illusions?
Um, do you think that is about faces or just generally visual processing in the brain?
It's this question from the different faces identifying questions, so I
Got it.
think it's
Okay.
kind of like related if that also applies to.
I don't know of specific illusions about
Oh, so
face
sorry, the
discrimination.
mental hallucinations.
Hallucinations. Hallucinations.
Yes, thank you for
Well,
clarifying.
certainly. Hallucinations, hallucinations make a lot more sense when you understand that the brain constructs your reality. And and dreaming is a sort of a kind of a hallucination, right? Like you don't you can have very vivid dreams that you are in a particular place or with a particular person identified by their face or just by something about them, right? And all these are patterns of neurons in your brain that would be a similar pattern of neurons that would be active if you were at that place or you were with that person looking at that person. Um, if a similar pattern is activated during a dream, or if you are having a hallucination, you're going to have a similar experience, a similar perception. And so I don't, again, this is a little bit outside of my expertise, but um, things like dreaming and hallucinations make much more sense when you understand how the brain operates under normal circumstances, because it's just a replication, a repetition of a particular pattern that while you're sleeping or when you're in an altered state of reality, that gives you a similar experience.
Great. And then I'm going to jump to this other question because I think it is somewhat related.
Mhm.
This question is, and then why are some people face blind? Um, yeah,
Yes. So yeah, face blind is really fascinating. Condition is called prosopagnosia. Um, and I'd mentioned Oliver Sacks, who is a neurologist, has written a lot of books. He is a prosopagnosic, and has written about the experience himself. Um, uh, and he's written about it in other people as well. And prosopagnosia, or face blindness, is caused by damage to the human version of IT cortex. It's called something different in humans. It's called the fusiform face area, but it's in pretty much the same place in humans as IT cortex is in monkeys. Um, and it's a place where the sort of neurology, if someone has a brain injury, if they have a stroke or a bullet wound to this part of their brain, and they lose the function that you could deduce by these single neuron recording studies in the monkey, and also the same similar studies have been done in scanners and with electrodes in people while they've had surgeries. So um, yes, face blindness is fascinating. And again, it ties back to this idea that a lot of what was early on learned about the map in the brain was from neurology. It was from people who had specific brain damage and somehow survived it. And then had, if it was specific enough, then you could learn, oh, this is where in the brain this function happens. And oh, the brain does it in this particular way because person has lost this function, but not that function. Um, and so prosopagnosia is yet another example of that, where if you know where the damage is, now you've done a one-off brain mapping study.
that's really interesting. Thank you. Okay, we have a couple more questions still.
Mhm.
This one is, will we be able to correlate spinal cord injury with changes in the mapping?
Yes. Um, that's the simple answer to a very interesting and complicated question. So, um, there are a couple aspects to this. So one is that um, if you have a spinal cord injury and you lose sensation from part of your body, or you lose the motor control of a particular part of your body, then um, that change in your experience of not being able to move or not getting sensory feedback will over time cause a remapping in your brain, because your brain is constantly adjusting and learning to its environment. And if you change something about your environment, your brain will remap itself. Um, that's one thing. Another thing is that and a sort of related thing is the concept of phantom limb. So if someone has an amputation, they lose their arm, your body had a representation of your arm. Sorry, your brain had a representation of your arm. You lose your arm, the representation in your brain doesn't go away immediately. And that is the experience of a phantom. You do not have an arm, but it feels like you have one. And sometimes people even have pain in this phantom that they can't do anything about because there's no way to treat it. Or at least it's not obvious to them how to treat it. There are some tricks. Um, but over time, the representation of the brain may change, and so that phantom may sort of telescope in and get shorter and shorter and eventually disappear, or it may not. Um, so changes to the rest of your body, whether it be a spinal cord injury or an amputation or a numbness caused by a disease, those will cause a remapping in your brain. And that's been seen in humans, and it's been seen in more systematic studies in animals. Um, and the last thing I'll say about that is that there's some really exciting work in which um, scientists are using robots to try to cause plasticity or changes in the spinal cord as a way of speeding up the recovery from a partial spinal cord injury. And this work first has to be done in animals. There have been these really interesting studies in rats and monkeys that I don't have time to go into right now, but they've actually now implemented these in humans as a result. So these are cases of animal studies of trying to treat spinal cord injury by causing plasticity or rewiring in the spinal cord or in the brain, using robots, and then applying the same thing that was done in animals to human patients who have an accidental spinal cord injury, and having some great success with that.
Thank you. Our next question is actually a bunch of questions kind of in one, but I think it's a pretty interesting one. Since there's so much variation, could brains be transplanted? And then how would that even work? And then in addition to that, could brains be grown one day to replace the brains the in like progressive Alzheimer's patients? And do you think it'd be ever possible to even duplicate memory or even duplicate a part of the brain?
Okay, these are fun, sort of science fiction level questions,
Yes.
mostly because these are not close to science reality right now. And there are a lot of very interesting sort of thought experiment questions and sort of ethical considerations. If you could get this technology, there's a lot of things you'd have to consider as to whether you would want to do this. So, um, can brains be transplanted? There have been studies in which, in animals, there was a head transplantation tried, and that's not the same as a brain transplantation exactly. But the idea is, could you reconnect the nerves and the vital connections of the heart and the lungs to the head, um, in order to keep a head alive, um, so that someone who had a failing torso could maintain their head that has their personality and their memories and their consciousness in it. And there's some very preliminary studies in animals that suggest that this is theoretically possible, but it's very difficult to do. And I think it's a long way from even considering that in humans. Now, um, an interesting thought experiment is, could you make a copy of a brain? And if you did, would it be the same person in that brain? Okay, so let's imagine that those connectomes I was talking about, that where right now the state of the art is that 100 people spend multiple years to make a connectome of 1 cubic millimeter of a mouse brain. Okay, let's say our technology increases rapidly and you could somehow do a connectome of a whole human brain, which at this point right now would take thousands of years, tens of thousands of years with our current technology. Um, if you could do that, then the next thing that would be even more difficult is, could you then reproduce every single one of those billions or trillions of connections, and not only reproduce all of those connections between all those neurons, but um, get the strength of all those connections to be identical? Those are a lot of really, really big ifs. Then, if you put that brain in somebody's body, would they have all the same memories? Would they be a copy of that person? Would they be that person? Would they have the consciousness of that person? Um, uh, that is certainly at the stage only of a thought experiment. Right now, it's not possible. You could sort of imagine how parts of that might be possible. Other parts, I can't even imagine how it would be possible to rebuild that. And in order to do a connectome like that right now, you need to chop up the brain into very, very thin pieces. So you would destroy one brain in order to potentially make a copy of it. So there's a lot of technical problems with that idea, but it's a really interesting idea to sort of play with in terms of thought experiments.
Yeah, that's very fun to think about, but also, yes, like you said, um, almost inconceivable how it would actually work. But
Yeah.
um, our last question here isn't actually a question. It's just a lovely comment that I want to share. I thought it'd be a nice way to wrap up.
Mhm.
Is just from one of the audience members saying, super interesting presentation and thank you. And I have to say the exact same thing. This was so fascinating. I feel so fortunate that part of my job is getting to learn from incredible people like yourself. So thank you to everyone who joined us this evening for this talk. And I've had a wonderful time. We did so great. We're right right at time, wrapping up perfectly. So thank you to everyone. We will be sending that follow-up email. So if you aren't on the list yet and you're interested in coming in the future, please join up the mailing list. As I mentioned, we are taking a little break for the summer, but we look forward to coming back in the fall and to more wonderful talks like tonight. So good night, everyone.
Thanks, Stevie. Thanks, everybody.